This is kind of like part 2 of Project 6, and just like but like setting sites a little lower, of doing a minimum viable upload search. So this is like looking for the smallest organism / cells / brain slice to fully upload and then prove that you've done it. So real cells, either on a chip or growing in a dish or swimming around. Criteria you'd use to search for this is low number of neurons. So something very simple. I mean, we already have C. elegans sort of as the model, so it's like looking for something better than C. elegans, right? Something simpler than that. Something more similar to human neurons, something much easier to work with, hopefully still transparent like C. elegans, but non-pressurized, so when you poke it, it doesn't goo out. And something maybe with more charisma, something cute, photogenic, thinking tardigrade, water bear sort of thing. That also has like less neurons, maybe not as transparent though. And then something that's easy to give it a memory and then test whether that was captured. So you can see like brain slice kind of doesn't really fit that, some of these other things, but it's like very similar to, it is human neurons. So this would be a search, small project, search for a better model organism. C. elegans is the thing to beat. What about rotifer? We just, on Wikipedia, list of animals, rotifer has 200 neurons. It's also transparent, has maybe 1,000 cells. Tardigrade, also 200 neurons, 1,000 cells. So that's like, 66% less than, C. elegans. So maybe it's easier. Yeah, and then some of the standard, I didn't really fill out some of the rest of the stuff, but the target is, you're trying to answer, you're lowering the barrier to answering the important questions such as, what is the capture resolution that preserves memory and allows for future learning of an uploaded organism? And I think that's pretty much all the things I wanted to mention. So it's kind of like a piece of like a subset of Project 6. I imagine this is kind of like one person for 6 months sort of level. With the 1 or 2K, it would mostly be like doing a literature review and finding like what other things have people already been looking at, and is there anything better, including looking at brain slices, and can you entrain memories in those and keep them alive for a while.
I wonder, there is some knowledge about selecting model animals in different parts of biology. A lot of it is the tradition. A lot of people are just using the animals, use it because there's a lot of literature about them. But I seem to have come across discussions about optimal model animal selection at some conferences. I never really paid much attention, being on the pure theory side myself. But there might be some neuro people out there that could point you in the direction of that, because there might be a way of getting good inputs on this.
And some things that they care about very often are which animals can do the most manipulations with DC, and that it's typically things where, for instance, you've already bred a bunch of mouse lines or rat lines, and you already know how to do optogenetics with them. And so that's why that comes up. And the same sort of thing with Drosophila. Drosophila has had a lot of research. So that's often an argument by neuroscientists, but I don't know if that's what has to apply here, because, you just kind of want to register all the neurons. And maybe you can do it with an array and you don't have to worry about all that. So, yeah.
The best thing for this is an all-optical human brain slice,
Yeah.
where you write engrams in with LTP and LTE explicitly as bits and read them out with voltage, dies in and scan the whole thing. And I wonder if anybody has thoughts on that or improvements on that, or knows about brain slices and might be able to talk to us later about this. Does anyone actually do brain slices here?
Well, the all-optical reading and writing was done to encode an engram by the Justa lab, and they actually screwed up that. And it turned out that they thought that they were encoding by synaptic strengthening, like you would normally do, but they were actually seeing an intrinsic difference to the neurons that looked like a engram, if you will.
Do you have any thoughts on how many bits we can write in? I've talked to some of the companies that purport to be able to do this, and it it definitely is kind of cutting edge and like you'd have to put it together, but I was thinking something on the order of 80 bits for a like couple millimeter square slice.
We should talk.
Yeah.
Okay, thank you so much, Michael.